Hello friends, welcome to Sam Biology class. Today we shall discuss about non-disjunction. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Non-disjunction is the failure of homologous chromosomes or sister chromatids to separate properly during the cell division. It is the leading cause of pregnancy loss and birth defects. The result of non-disjunction is aneuploidy, which refers to the presence of abnormal number of chromosomes in a cell. Each type of aneuploidy can be attributed to non-disjunction during mitosis or meiosis. Kelvin Bridges and Thomas Hunt Morgan are credited with discovering non-disjunction in Drosophila melanogaster sex chromosomes in 1910. There are three forms of non-disjunction. One is non-disjunction in mitosis, number 2 non-disjunction in meiosis 1, and number 3 non-disjunction in meiosis 2. There are two parts to the cell cycle. One is interphase, and the other one is mitosis or meiosis. Interphase is further subdivided into three phases. They are G1 phase, S phase, and G2 phase. In G1 and G2 phase, cells grow in size by producing various proteins. G phase is the growth phase, whereas in the S or synthesis phase, DNA is replicated, as a result of which each chromosome includes two identical sister chromatids. This figure describes mitotic non-disjunction. Mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. In this figure, this is a diploid cell having two pairs of chromosomes. So totally the cell has 4 chromosomes, and each chromosome consists of two identical sister chromatids. In prophase, nuclear envelope breaks down and chromatins condense. In metaphase, chromosomes are lined up at the metaphase plate and microtubules are attached to the kinetochore of each chromosome. It is the microtubules which pull up the sister chromatids to opposite pole. Metaphase is followed by anaphase, where the sister chromatids separate and move to opposite pole. Anaphase is followed by telophase, where nuclear envelope reappear and chromosomes decondense into chromatin. This is followed by cytokinesis, in which the cell is divided into two daughter cells. In this figure, the sister chromatids of chromosome number 3, they fail to separate and both sister chromatids move to one pole. And in the resulting daughter cells, one cell will have 5 chromosomes and the other cell is having only 3 chromosomes. So mitotic non-disjunction results in the formation of two daughter cells with abnormal number of chromosomes. This is due to the failure of separation of sister chromatids in anaphase. In this example, one of the daughter cell will have one extra chromosome. So it will have 2n plus 1 is equal to 5 chromosomes, whereas in the other daughter cell, one chromosome is missing, because of which this chromosome will have 2n-1 is equal to 3 chromosomes. Mitotic non-disjunction can arise due to the inactivation of either topoisomerase 2 or condensin or separase. It gives rise to somatic mosaicism in which the abnormal number of chromosomes are observed in the cells which are the direct offspring of the cell where non-disjunction had occurred. It gives rise to certain form of cancer including retinoblastoma. In addition, non-disjunction can occur during either Meiosis 1 or Meiosis 2 with differing results. This figure describes the outcome of non-disjunction in Meiosis 1. Non-disjunction in Meiosis 1 occurs when the pairs of homologous chromosomes or the tetrads fail to separate in anaphase. Because of this, out of two daughter cells that are formed at the end of Meiosis 1, one daughter cell will have n+1 chromosome and the other daughter cell will have n-1 chromosomes. In this figure, you can observe that there is close pairing between homologous chromosomes. This pairing is known as synapsis. Because of this pairing, exchange of genetic material takes place between the non-sister chromatids of homologous chromosomes. This is known as crossing over. So in this figure, it is due to non-disjunction both the homologous chromosomes move to one pole. Because of this, when two daughter cells are formed, only one daughter cell will have this pair of homologous chromosomes, whereas in the other daughter cell, this pair will be completely absent. The daughter cell which has this pair of homologous chromosomes will have n+1 chromosomes, and the other daughter cell from which this pair is missing will have n-1 chromosomes. When these two daughter cells divide once more in Meiosis 2, 4 daughter cells are formed, out of which 2 will have n+1 chromosomes and the remaining 2 will have n-1. So friends, this is about non-disjunction in Meiosis 1 that takes place due to the failure of separation of bivalents in anaphase. Non-disjunction in Meiosis 2 occurs due to the failure of sister chromatids to separate properly in anaphase. As Meiosis 1 proceeds error-free, the two daughter cells that are formed at the end of Meiosis 1 are normal, having n chromosome. But in Meiosis 2, non-disjunction takes place. As a result, sister chromatids fail to separate, that results in aneuploidy. In this example, this daughter cells divides normally without non-disjunction. So the chromosomes are equally distributed between these two cells. So the resulting daughter cells are normal, having n chromosomes. In the other daughter cell, which is formed at the end of Meiosis 1, these two sister chromatids, they fail to separate. So both of them move to one pole, whereas the other chromosome is equally distributed. When the cell divides in Meiosis 2, out of two daughter cells, one cell will have n-1 chromosome and the other cell will have n+1 chromosome. Because this pair of sister chromatids remain in one daughter cell, so it will have n+1 chromosome. Whereas in the other daughter cell from which the sister chromatid is missing will have n-1 chromosome. So friends, non-disjunction in meiosis II occurs due to the failure of sister chromatids to separate properly in anaphase. Now the question is why homologous chromosomes or sister chromatids fail to separate properly. Let us discuss the molecular mechanism of non-disjunction. First, what is spindle assembly checkpoint? It is a molecular mechanism that governs proper chromosome segregation in eukaryotes. This checkpoint inhibits progression into anaphase until all homologous chromosomes are properly aligned to the spindle apparatus. Then only SAC releases its inhibition of anaphase promoting complex, which in turn triggers irreversible progression through anaphase. Another important point is sex-specific differences in meiosis. Survey of cases of human aneuploid syndromes have shown that most of them are maternally derived. The most notable difference between oogenesis in female and spermatogenesis in male is the prolonged arrest of oocyte in the late stages of prophase 1 for some years. In contrast to this, male gametes proceeds quickly through all stages of meiosis 1 and meiosis 2. Another difference between male and female meiosis is the frequency of recombination between homologous chromosomes. In male, almost all chromosome pairs are joined by at least one crossover, whereas more than 10% of oocytes have at least one bivalent, which is without any crossover event. So failure of recombination and inappropriately placed crossover are one of the major contributors to the non-disjunction in human being. Another important point is the age-related loss of cohesin ties that hold sister chromatids together is one of the contributors to the occurrence of non-disjunction. So friends, this is all about non-disjunction. For more updates, please subscribe my YouTube channel, Sam Biology Class.